My name's Nynke. I'm currently doing my PhD on Tasmanian devils, and I wanted to share some of the results that I've got so far. So this is a Tasmanian devil, for everyone who doesn't know. And Tasmanian devils are a carnivorous marsupial that is native to Tasmania, which is in the southern bit of Australia. And they have a disease called devil facial tumour disease. So this poor little guy here has a tumour on his face, that's DFTD. DFTD was first detected in 1996. It's a transmissible cancer, so that means that the cancer itself is the infectious agent, and so the cancer cells themselves are passed from devil to devil. It's generally fatal for the animals, and it means that in the wild, the devils before DFTD used to live maybe 4 or 6 years. Now most of them live 2, maybe 3 years, and they have fewer reproductive events. And the combination of the DFTD fatalities means that the population has declined by around about 80%. However, it's not all bad news. The research that's been going on has identified changes in several different cancer-associated genes in quite a short period of time, only a few generations. There is evidence that the devils themselves are surviving longer with the visible tumours. And this is a DFTD tumour that is detected in an animal that was captured in May 2018. This is the same animal captured 3 months later. You can see that the tumour is completely disappeared, which is really exciting. And so there has been evidence of tumour regression in multiple devils, and actually initially it was more common in female devils. We don't know why that is. And despite the research, there's actually still quite a bit about the devil's immune system and things that affect the immune system that we don't understand. So my hypothesis is that immune gene expression will be influenced by many different factors. So the first factor that I'm going to look at is season, and the vertebrate literature usually has differences sort of between summer and winter, or between breeding and non-breeding seasons. There can also be differences in geographic location, and this is often cited as things to do with climate or altitude or the local microbial community, which we've heard a lot about today. As I mentioned, we already know there are some sort of sex differences within the devil immune system or immune function, and we know from human studies that there are sex differences in human immune functions. We also know that age can affect how well the immune system functions, and previous research in the devils has shown changes in gene expression with age, specifically to do with the difference between immature and mature devils. And so all of these factors could potentially alter the devil's capacity to respond to DFTD infection. So I wanted to sort of get an overview of the devil immune system, and I looked at the expression of several receptors that are on different cells associated with either the adaptive or innate immune system. And I looked at their expression in association with DFTD, location, sex, age, and season. So I have samples from 4 different locations. So this location here, Helena, is where DFTD was first detected in 1996. West Pencil Pine is a location, a population that's been quite heavily studied, and there's a lot of early literature that was based in this population, and so there's quite a lot of knowledge regarding the animals that live here. Tacone is the location where the regressions were first detected, and Black River is a location that has only had the DFTD infection present for about 4 years or so. And the samples in the study were collected between the end of November 2016 and mid-2019, they're blood samples. So I'm examining circulating lymphocytes, and there's 380 individuals. So RNA was extracted and checked for quality and quantity, and qPCR was run on a selection of 10 genes. The CT values were then standardized using a FAFL method. All of the statistics was done in R because some of the genes had quite high correlations. It was decided that we should analyze the genes separately. So all the variables for each gene was put into a model selection analysis, and those variables that were deemed most important were included in the final linear models, and all statistically significant results, their marginal means were extracted and they were visualized. So this is my results. Let me explain what this is. So the heat map represents the scaled model slopes for each of the models. Each column is a separate gene model, and each row is a separate explanatory variable or an interaction between two explanatory variables. The colors are representing, the warm colors are positive scaled slopes, and the cool colors are negative scaled slopes, and then the asterisks are significant, so p-values. And I mean, what you can see initially is that there are quite a lot of different variables that are affecting the different genes. I'm not going to talk about all of them. That would take a long time. There's two that I really want to talk about. And the first one is this sort of orangey red patch here, which is the interaction between season and location. It was significant in about 3/4 of the genes. The other thing I want to talk about, I don't know if you can see, but this row here is the main effect of DFTD. And so most of the genes in the study were downregulated when the devils have DFTD, with the exception of this gene here, which was strongly upregulated. So let me show you what this looks like. So this is a location by season effect. I've chosen CD4 to show you, but the patterns were quite similar across the different genes. And one of the things that I want to point out is that West pencil pine is here in the red, and it's quite different in terms of its gene expression compared to all of the other locations. So the other locations are essentially showing no strong seasonality. They're not really showing any difference, whereas West pencil pine has this strong downregulation in both autumn and spring. This was a bit of a surprise, and at the moment I don't really know what's causing these downregulations. My current theories is that West pencil pine is a lot colder than the other locations, and there might be something in the unique climate. Of course, it also could be something to do with a unique pathogen community in that location. The other result I wanted to talk about was CD16. So that's the other name for CD16. Some people might know it that way. But the first thing I want to point out is that if you look just at the healthy devils, West pencil pine again is significantly different, and it's downregulated compared to the other locations. And the other thing, of course, that's really interesting is this gene is upregulated with DFTD infection. Now, this is gene expression, and CD16 is mostly expressed on natural killer cells, monocytes, or neutrophils, and I don't know which one of these genes is driving the expression difference. I would love to find out and do more research on that in the future. I also don't know if this association is an immune response by the host or something that the tumor is modulating for the host, or simply a byproduct of some sort of physiological changes that are occurring when animals are infected with DFTD. There's a lot that's not understood about the mechanisms, but it's still really interesting. So in summary, there are geographical and seasonal influences or variations on devil immune gene expression. And this is important for conservation management because it means that there may be unique differences within the populations. And so when you make decisions about the whole population, you really need to consider multiple locations. The association with CD16 expression and DFTD is interesting, and it highlights the importance of the innate immune system in this system. So the next steps for my PhD are to look at climate, add that to the analysis to look more closely at the pathogen community. I have some longitudinal samples which I need to finish analysing, and I want to look at tumour growth rates and immune capacity. And so with that, I would like to thank my supervisor, Dr. Ujafari, and my lab group, the Wild Genes group, and all the funding partners. And of course, I would love to thank the Tasmanian Devils.